Organoids are the closest structures to our organs that we can make in the lab. You can imagine them as miniaturized and simplified versions of our organs, if you will. But they're not fully functional yet, which means we can't transplant them to the patients, and we have a long way to make a complete organ in the lab. But people always ask, we discover stem cells and they can make all of the cells in our body, so give me my organs. I wish, but it's not that simple, and we achieved this technology just a couple of years ago. The other question is that if they are not functional for transplantation, what's the point of making them? Traditionally, if we wanted to deal with a disease or study some biological events in our body, our options were to treat people with uncertainty, or we could experiment on animals. So it would be great if we had a model for studying and testing new treatments to make sure they would work in individual subjects. Besides, making animal models with a specific disease is difficult, and there's a huge difference between animals and humans. Some drugs may work on animals, but not on humans. Even if we just talk about humans only, the individual difference can also affect the treatment. So if we could somehow isolate the stem cells from a patient, we can make organoids specific to that patient, and we can experiment and study them in the lab. So in this case, the data is quite helpful to make and test new treatments, even though we are still trying to complete our mission to make functional organs for transplantation. But I think it is interesting to know how exactly a cell could turn into an organoid and what stops us to make a complete and transplantable organ. 3 different discoveries have been made in the late 19th century, and their advancement help us to make such complex structures in the lab. The first thing you should know is the level of organization in our body, which I probably repeated hundreds of times in previous videos, but a variety of cells with different shape and sizes make our organs. They have specific functions in our body, and we call them fully differentiated cells. They are in a state that they are not capable of cell division, which we call G0, resting or quiescent phase. On the other hand, we have stem cells that are in the G0 state, but with proper signals, these stem cells can exit the resting phase and start to grow at interphase and divide with mitosis, which we can describe with the word proliferation. So stem cells can make more of themselves with cell division, and with the specific signals, their gene expression can be changed and differentiated into more specialized cells with different shapes, sizes, and functions. Those specialized cells can do the same thing and make even more specified cells. So they can make a lineage of cells until they produce fully differentiated cells. Therefore, the number of fully differentiated cell types is related to a stem cell's capability to how many types of cells they can make. So we have different types of stem cells with different potential. For example, pluripotent stem cells can make almost all of the cells in the human body, but unipotent stem cells can only give rise to just one specialized cell type. Let's imagine that you isolated a bunch of cells from your body. After the isolation, the cells need a place that mimics their natural environment. The first cell culture was reported by Ross Harrison in 1906, and he wanted to observe nerve cells under the microscope. He isolated an embryonic tissue and observed its differentiation towards nerve fibers, and he managed to keep them alive for nearly 4 weeks. But this was the interesting part. He has stated that the cells do not undergo normal transformation because of the abnormal conditions of the mechanical tension to which the cells are subjected. And this is completely accurate because this method of cell culture does not mimic our natural biological systems. In a 2D cell culture, we use a flask or petri-dish and a medium which consists of the necessary ingredients that our cells need. In a petri-dish, the cells can attach to a 2-dimensional plate with X and Y axis, and they can have limited 2D contact with other cells, which is already a huge change from their natural environment. If we use a non-adherent plate and increase cell-cell interaction with different methods such as using a hang and drop plate, the cells can attach to one another and make a spheroid. Actually, this is the method that Madeline Lancaster used to start making an organoid in this paper, which I want to talk about in this video. But to understand this, first you need to understand that all of our cells in our body are made from a single cell called zygote, which is a combination of a sperm and a single egg cell in the fertilization process. Then with a lot of cell division, it makes structures that we call blastocyte, which has a type of pluripotent stem cells called embryonic stem cells. And after that, they start differentiating and making 3 different layers of cells: endoderm, mesoderm, and ectoderm. Each layer gives rise to specific tissues and organs in the developing embryo. So embryonic stem cells can make all of the cells in our body, but we can't use these cells to make organoids. First of all, it raises ethical problems because by isolating these cells, practically we would kill the baby who would be created by these cells. And if we want to make a disease model or a specific organs for a specific patient, we need to use another pluripotent stem cells. In 2006, Shinya Yamanaka found that if we add certain signaling molecules to the medium, the fully differentiated cells can be reprogrammed to pluripotent stem cells. These induced pluripotent stem cells have similar properties compared to embryonic stem cells. So if our fully differentiated cells have some mutations which cause some specific conditions, the induced pluripotent stem cells have the same mutations as well. And if we can make an organoid with these cells, we can have an accurate disease model that we can run a variety of tests before the actual treatment for that specific patient. To make a brain organoid, Lancaster and her colleague isolated the skin cells from a patient which had microcephaly. Then they reprogrammed the differentiated cells to induce pluripotent stem cells. Afterward, they cultured those induced pluripotent stem cells on a non-adherent plate, which helped them to make spheroids. The stem cells in spheroids start differentiating to make the 3 germ layers, which we call these structures in braid bodies. If you do nothing, they continue differentiating and make many fully differentiated cells like intestine cells or kidney cells or hair cells. In this paper, they put inbred bodies in a medium that doesn't support the mesoderm and endoderm, so eventually they die out and the ectoderm remains because we need it for making our nervous system. After that, they put the inbred bodies with only the neuroectoderm layer to the droplet of a matrigel and they put them in a differentiation media. Okay, what is the matrigel? The matrigel is a product name that acts as a 3D scaffold for the cells and scientists use them for 3D culturing the cells. It's rich in 2 types of proteins that can be found in a natural extracellular matrix, which are deposited by specific mouse tumor cells called Engelbreth-Holmes swarm. For making an organ, different cells and tissues should position themselves in the right place to be functional. For example, if you look at every skin structures, you can see different layers of cells in the exact position. So how we can make this complex cell sorting in our experiment? Actually, we don't need to do anything. The cells know exactly how they should gather together in the right place. This was explained by differential adhesion hypothesis, which proposed that the cells rearrange themselves in positions with more thermodynamically stable structures. So what does that mean? Imagine you have 3 different layers of cells organized with the specific orders, and you put them in an alkaline solution to dissociate them into individual cells. Here's the interesting part. If you mix them together, they would reorganize in the exact position as before. Different cells together make cell-cell interaction with the specific proteins on their surface, so they love to rearrange their position with the lowest interfacial free energy. So in this paper, they just made an organoid with the minimum interventions possible. They just made a 3D environment with the specific signals and they let the cells do the rest.
To take this approach and basically just sort of support the tissue, allow it to do what it's already programmed to do with as minimal intervention as possible. And so
after they moved the neuroectoderm to the matrigel, the cells reorganized perfectly in the gel. And after 2 months on an orbital shaker in a differentiation medium, you can have a cerebral organoid up to 4 mm in diameter. So this is it for this video and thank you for watching.